mrvp broth was developed in 1915 to distinguish between the large number of fermentative enteric bacteria that inhabit the human intestine. mrvp broth is a glucose broth used for two tests. MR, or methyl red, is used to detect the production of large amounts of lactic acetic and formic acid produced during the fermentation of glucose. Methyl red turns yellow at a pH below 4.4. VP stands for the Voges-Proskauer test. All bacteria do not produce acids from fermentation. The VP reagents detect the production of a 3-carbon alcohol called acetoin. Label 2 mrvp tubes for each organism to be tested. We will be working with Proteus vulgaris, but the organism used in your lab may be different. Obtain an inoculum using a sterile inoculating loop and inoculate the 2 tubes. Flame the loop and let it cool. Use the loop to obtain a loopful of bacteria and inoculate the first tube. Flame your loop, let it cool, and repeat the procedure to inoculate the second tube. After you've inoculated the second tube, flame your loop again. Incubate both tubes at 35 to 37 degrees Celsius until the tubes have been incubated long enough for the bacterium to fully catabolize the substrate sugar, usually 48 hours. Prepare controls by pouring half the contents of an uninoculated mrvp broth into a clean tube. Keep the remaining uninoculated mrvp broth for the VP control. Label the tubes. Label one tube MR control and label the second tube VP control. Add 5 drops of methyl red to one inoculated tube and to one control tube. The number of drops will depend on the volume in the tube. Hold the tube at an angle and let the methyl red drip down the inside of the tube. Observe for a color change to red. The Proteus produced enough acid to change the color of the indicator to red. Add 12 drops of VP reagent 1, alpha-naphthol, and 3 drops of VP reagent 2, potassium hydroxide, to one inoculated tube and to one control tube. Cover each tube with a parafilm square. To cover the tubes with parafilm, use scissors to cut a small square of parafilm. Remove the backing from the parafilm and put the parafilm over the top of the tube. Stretch the parafilm down towards the base of the tube. As you stretch the parafilm, begin to wrap it around the tube. Smooth the parafilm so that it sticks securely and provides a tight cover. Carefully shake the tubes side to side, not up and down. You can also use a vortex set to low to mix the contents of the tubes. Remove the parafilm from the tubes and discard the parafilm in a biohazard bag. Leave the caps off the tubes and let them stand for up to 30 minutes. If bacteria produce acetoin, a red color will develop at the top of the broth. Remove the labels and dispose of the tubes properly.